So this is stem cells and cancers. And cancer cells are basically stem cells, and they do share pretty much every hallmarks of cancers. And it is further evidenced by the high correlations of the stem cell division and cancer incidence. So one is basically 100% correlations, and they are basically like 100% correlated, but so there's a high correlation between the number of stem cell division and cancer incidence. And this shows the red is 100% and white is 0%, and there's a very low percentage of the hereditary mutations in women with cancer, or there's a hereditary cause of cancer. You're like destined to get cancer, but it's pretty low. And there's a replicative, so it's like as the cells replicate this, when they usually accumulate the DNA damage and cause cancer. There's some environmental, such as like skin cells and lungs. There's a pretty big environmental causes to the cancer. So this is preventable, but the other two are not really preventable. And the stem cells, when they divide into two cells, they have one cell just as the same stem cells, which is self-renewal. Then they be divided into progenitors, and then progenitors become differentiated cells. And yeah, and they keep these cycles so that they can continue to like make more cells. So that's why they have this stem cells. So we have the word pluripotency, and which means it just can be anything, any type of cells. There are different type of potency. Totipotent is it can literally be anything, even like umbilical cord. And pluripotent cells is pretty much anything in our body. And multipotent is like adult stem cells. Unlike the other two, were found in like pretty much like fetus in the womb. Now this is found in adults. But it can still be many things within, as long as it's within the group, like skin or like brain, etc. It cannot be both brain and blood. Okay. So we have this ESL, which have blastocyst, and it has an inner cell mass, which is like one of the most potent stem cells, and they can literally make anything from brain to liver to etc. Then when it becomes embryo, we can not just no longer get like this tail and then make it into a neuron. We have to get this gonadal cells, and that still has a lot of potential as a stem cells. It can be many things. And if it's an adult, then we have to get the spermatogonial stem cells, but it can no longer be anything anymore. It has to be in a specific like same like group. And then we have this. We can also do to make the pluripotent stem cells, we can induce them, so which is what this I stand for iPSC cells, and we put these Yamanaka factors to go make them de-differentiate, and yeah. It's a hematopoietic lineage, so the stem cells can be like many type of different blood cells, basically. And so this is a pyramid of their potentials or what they can be. And so they start from totipotent, pluripotent, multipotent, and then they commit to like neuronal. They're like neuron, and then they can further commit to glial type cells, and then they have terminal differentiation. Here the word bivalent means it has a pair of gene, but one of them is repressive and another is active, so it's like an in-between of active and repressed state. So it's like same figures above, but this is a little more realistic. So as the stem cells can give rise to another same stem cells or become differentiated cells. However, differentiated cells can sometimes, in certain occasions, de-differentiate into stem cells. Also, stem cell division goes as shown. Stem cell give rise to restricted potential stem cell, and that give rise to progenitor cells and then terminally differentiated cells. And what this blue arrow is showing is that it can self-renew, so makes two daughter cells, where one is original and the another is like a new type of cells. And most cancer cells are progenitor cells, which means they can keep dividing with this self-renew cycle. And terminally differentiated cells mean it's done dividing, and all there's left for is to die. And we have two theories on how or why the stem cells become a cancer. One is the stem cell leads to a cancer cell by replicating and accumulating mutations, which makes sense because they go through a lot of division. They have a lot of opportunities to accumulate the DNA damage, as shown in the correlations between the number of stem cell division and the cancer incidence on this graph. And then the second theory is that progenitor cells can de-differentiate to become stem cell due to the damage that they have occurred or the mutation that they have. So a lot of mutations / DNA damage somehow cause progenitors to de-differentiate, which normal cells don't. And then once it becomes stem cells, it's already a broken DNA, so it's gonna become cancer. So this is how usual cancer looks. In the core, there's a cancer stem cells, and then they're surrounded by cancer cells, and then there's normal cells. EMT is basically like cancer cells. It's no longer like fixed in a place with other cells. Instead of being a happy neighbor, they get angry and then go to like different places and start spreading cancer. And they're just more tolerable to hypoxia and drug afflux, so they're more drug resistant. The stem cells, they basically live forever, so they have more mechanism to keep them alive. And cancer stem cells, of course, there's a part of the stem cells that they are also have a stronger survival instinct and more resistant to drug as a result. And cancer cells, so cancer is really fueled by cancer stem cells. Even if you put cancer cells, regular cancer cells in there, like a different animal, it doesn't really lead to cancer. But if you put cancer stem cells, it will lead to cancer. And this is just a chart that shows how resistant cancer cells are to hypoxia. For example, some of them are 20 times more resistant to oxygen deprivation. So in like a GI tract, there's like normal stem cells, and then some days like slowly they become more and more like mutated, and to a point where they become cancer stem cells, and they're fully de-differentiated, and it's a cancer, and it starts to spread to other places. So we have like this a lot of surface area, and that's to absorb nutrients in the GI tract. And APC, which is on amplifying cells, which we'll talk about later, or here. What these do is I'll talk about it later.
I don't know, actually.
So deletion of APCs in transit, here it does not cause tumor, but deletion of APC in stem cells over here will cause tumor. And so we know that APC plays an important role in cancer carcinogenesis. So what APC does is that it usually like limits the production of this or activation of beta catenin, but in the absence of APC, it will be produced and it will make more cells. So it's basically cancer or tumor suppressor gene. So similar to shown, there's a CS cancer stem cells and then surrounded by regular cancer cells. But CSC is what's really driving the cancer, and there's a Wnt signaling pathway, and so there's this frizzled receptor, there's an LRP, and then there's this like 4 proteins, and then if they exist, they cause beta catenin to turn into a proteosomes, and they degrade, and then no expression. But so they are destroyed without the wind, but with the wind, they get activated and they start expressing their target genes, and more production of genes or expression of genes, tumor. And so if you look at it,
I already have my doubts.
it's aimed for the beta-catenin, and then with the compared to normal cell, abnormal cells definitely has a lot more beta-catenin. And so what we can do to make drug is we have this molecule that they disrupts the binding of beta-catenin and TCF, which can disrupt the self-renewal cycle and cause differentiation. Once you're differentiated, you can no longer divide, and so prevents tumor. There are many different mechanisms that can prevent this Wnt signaling pathway. We have different one, the hedgehog signaling pathway. So when there is no hedgehog, then there's like really nothing really expressed. But when there is, the smoothened receptor can migrate into like this place, and they can break down the SUFU and PKA, which release the glee, and the glee then work as a transcriptor to express the target genes, which can be oncogene, such as like cyclin D, BCL, L2, VEGF, SNL, etc. So in order to inhibit this hedgehog pathway, we can disrupt it in multiple ways, as we can see here. So gene expression, regulation of stem cell differentiated by gene expression. There's a polycomb group PCG proteins. They're called guardian of stemness. What they do is they suppress the differentiation of related transcription factors. So all these are differentiation transcriptional factor. And what they do is this protein polycomb groups are suppressing the differentiation transcription factor, and maintain the stem cells. So they suppress differentiation from a self-renewal of the stem cells. So if you're stuck in one of this stage, or when something goes wrong, then you're like stuck in to become a stem cells and you get cancer. Leukemia, if there's a retonic acid, if there's no retonic acid, then it's under-differentiated and cells becomes a cancer. But let's force them to differentiate. That could be the therapeutic approach. So we can put, if there is retonic acid, then they like bring different coactivator, and then that allows it to express the differentiation of target gene. And if there's not enough retonic acid, it may not occur. So what you can do is you just put more retonic acid to increase the rate of differentiation. But actually you need more than just RA, you also need arsenic trioxide. But as long as you put that in, it kind of works, so that's great. And so APC is essential protein in the Wnt process, and they can break down the beta-catenin cofactor. And what that does is promote cell growth. So you're an inhibitor of the cell growth, you're an anti-tumor, tumor suppressing gene, right? And so by restoring the APC, you are able to control the growth of tissues. So this is like regular, and it looks fine. And this is also this is like there's no APC here, so it grows like everywhere. But when you like restore the APC, like it kind of looks normal now.